All right, so my presentation will have a lot of different scientific terms that you might not understand, but don't worry, I'll try my best to explain them. So a while back, I read a book in middle school, actually, 7th grade. And actually, I didn't really read that book because that book looks something like this. Yeah, you take one glance at it and you see there are many complicated terms, it looks very boring, and it looks very annoying to read. But I started reading it anyways, and after 2 pages, I was done. Instead, I decided to skim the ideas of the book to see what I can find. So basically, this book was titled Micrographia and was written by a scientist named Robert Hooke. And basically, what he summarized in the book was that he looked under a microscope, hence the name Micrographia, and described what he saw. And what he saw were bugs. But because he was the first person to see what bugs look like up close, he had the liberty to describe them for however he wanted to. So, when he looked under the microscope and looked at a flea, he described it as a little soldier, adorned with curiosity, polished suit of fable armor, awaiting his next journey. And if you take a picture, look at this picture of the flea from that book, he's not wrong. And the flea looks kind of daunting, and he described it in such a good light, which made the flea seem like a good thing, which the flea is not a good thing. But he had no choice but to describe it for however he wanted to. So next, he looked at a tree cork. Same concept, he put that tree cork under that microscope and took a look at it. And this is what he saw. Pretty boring, right? Cells, he called them, because they reminded him of the box-like cell structures and monasteries that he went to. And to this day, that is still what we call them, cells. Not because they look like boxes anymore, well, kinda, but because we like the name, I guess. But just like Hooke, we are still discovering new ideas, new concepts, and new things that we have never seen before. So, what do we do when we see new things? For the discovery of stem cells, it was promising, surprising, but not well received. Much like in vitro fertilization, which is the birth of a baby through laboratory procedures, it was challenged financially, politically, ethically, and medically until the birth of a healthy child. Instead of encouraging research that should be explored, critical research is challenged. But I'm not here to talk to you about human embryonic stem cells, which is the heart of the stem cell debate. I'm here to talk to you about a lesser known field known as human adipose stem cells, which has lesser ethical concerns than human embryonic stem cells. A huge misconception with ideas nowadays is that if one aspect of an idea is controversial, then the entire idea itself becomes controversial, and we kind of put a label on it. But it's important that we do not judge a book by its cover, and instead investigate that lesser known topic to see what light it can possess. So for stem cells, it is important for me to first describe what a stem cell is, as it is a very broad topic. So a stem cell is a specialized type of cell that can then turn into different types of cells. And it does this through 2 different procedures, self-renewal and differentiation. Self-renewal refers to the ability of stem cells to divide and reproduce multiple of itself. So you can see how that stem cell is reproducing into a different stem cell, and so on and so forth, indefinitely. Well, kinda. And then the second type is known as differentiation, as this is when the stem cell can turn into different types of cells. So such as fat cells, bone cells, nerve cells, and muscle cells. And again, there are many different types of stem cells, the most controversial one, again, being human embryonic stem cells. But a more interesting form of stem cells, in my opinion, are adult stem cells, specifically human adipose stem cells. So what are human adipose stem cells? Well, human adipose stem cells are stem cells taken from the adipose, which is the fat of the human body. And then these fat stem cells can then be turned into different types of cells, such as bone marrow, nerve cells, and muscle cells. And the first unique identity that human adipose stem cells possess is their location and ease of isolation. Because human adipose stem cells are taken from the fat of the human body, it does not pose any harm to the body or the area that it is taken from. We all have fat. Removing a small chunk of fat from that area does not overall harm the area of isolation. And this can be done through very minimally invasive procedures such as liposuction, making human adipose stem cells easier to obtain than other types of stem cells. And this is very important for forms of treatment to have this type of ability to remove from an area without damaging the surrounding organs because of removing the risk of donor site complications. So for example, lymphedema is a condition in which the arms or the legs begin to swell because of the blockage of fluid. And 25,000 to 50,000 new cases of lymphedema are found every single year. And there are only 2 current types of treatment for lymphedema, both of which are far from ideal and are very expensive, especially for people who cannot afford them. So the first type of treatment for lymphedema is lymph-venous bypass. And this is basically applicable for the earlier stages of lymphedema, but does not work for later stages and does not have a huge success rate either. And the second form is vascularized lymph node transfer, which does work for later stages of the condition, but comes with a huge risk, which is donor site complication. So basically, let's say I have lymphedema and I have a very severe form of lymphedema, and I have it in my arm, which is the area of swelling. So I need to remove the area of swelling. So I take a lymph node from my armpit, and then I take it and I implant it in my arm. Well, my arm may be reduced of the swelling, but then the area that I took it from may now be susceptible to disease and infection. So that is the risk of donor site complication. And that is in many types of forms of treatment, it has that type of risk. So when it comes to this, we need to find forms that does not require the removal of donor site organs. And human adipose stem cells does provide that potential. For example, I'm currently working as a local intern for a local hospital in which I'm trying to examine how human adipose stem cells can be implemented with a matrix to form a kind of niche that can be directly injected into areas of swelling or infection. So if you're directly injecting into that area, you are not removing other organs. And you can see how we have some kind of success with that. If you take a look at the diagram on the left side, you can see how those are basically lymphatic cells just by themselves. You can see how they're growing, as cells do, they just grow. But as soon as we add human adipose stem cells on the right side of the picture, you can see how straight from day 7, they are already forming like vessel-like structures. Because this is tissue engineering, we need to want to mimic the type of structure as close as we can. So vessels mimic veins and blood arteries, and you can see how that is having huge success because human adipose stem cells provide that. But why can't it do this in the first place? Well, this brings us to the second point, which is the regenerative properties and the pro-angiogenic growth factors of human adipose stem cells. So angiogenesis refers to the ability to develop new blood vessels. So pro-angiogenic growth factors are basically factors that help the development of new blood cells. And because human adipose stem cells are so abundant throughout the body, again, because we all have fat and that is the most abundant tissue, a smaller number of human adipose stem cells can produce a larger population of functional cells. So you can take maybe a small population of human adipose stem cells and then create a lot of nerve cells or a lot of muscle cells, thus making human adipose stem cells more effective forms of treatment. But why can't they do this in the first place? Well, just in general, stem cells are known as tropic cells, which means they are very happy cells. They love to reproduce, they love to differentiate, and they love to divide. So they're kind of always like that friend who's always trying to see things in a positive light. It's like, dude, I just failed this test. Well, don't worry, I'm sure you did fine. That's what stem cells are. No matter the environment around them, they're always trying to reproduce and see things in a positive light. So for human adipose stem cells, they do this through secreting a bunch of growth factors. So as soon as they're injected into an area of swelling and white blood cells are fighting for their lives, they're just happy to be there. So when they do that, they kind of just go, I secrete BFGF, I secrete another growth factor, and I secrete another growth factor. And then basically this all promotes the development of new blood cells, which is angiogenesis. And then overall, these factors all help, again, with tissue regeneration. And you have to remember that this is just the baseline of what we know so far. We are still in the preliminary stages of research for human adipose stem cells because, again, critical research is challenged. Just imagine the implications if it was encouraged instead. It goes beyond just medical treatments, but into economic remedies as well. So in terms of chronic wound healing, over $20 billion is spent annually on the treatment for chronic wound healing, and $30,000 to $50,000 is spent on diabetic foot ulcers in the US, which is the most common form of chronic wound. And in terms of lymphedema, 3 to 5 million people annually have this condition. And with current treatments far from ideal, human adipose stem cells can only help this. So continuing on the talk of chronic wound healing, human adipose stem cells can actually help this process. So inflammation is a very important healing process for any wound you have, but chronic inflammation can actually stop this process, which then can lead to a chronic wound, which can then lead to underlying conditions. So when implementing human adipose stem cells, that can actually block that procedure from the formation of a chronic wound, and instead regulate the immune response, which then leads to tissue regeneration. But of course, there are also clinical studies that have proven this. And in the first study above, a randomized trial of human adipose stem cells has been proven to be more effective in diabetic foot ulcers than standard treatments. And in the bottom study over here, another form of treatment with human adipose stem cells have been shown to be more effective in venous leg ulcer treatment. So again, you can see how clinical trials have proven that human adipose stem cells do have success. But then there are not without limitations of human adipose stem cell research. Again, this is only in the preliminary stages as we have not advanced further into this type of field, probably because the stereotypes that have been placed on stem cells as a whole. There have been many safety concerns with human adipose stem cells in larger clinical trials that still need to be conducted. But nonetheless, the potential of human adipose stem cells is still very encouraging. Even with the base preliminary research, human adipose stem cells have been shown to be more unique than other types of stem cells. So with the right mindset and with the right encouragement, human adipose stem cells can truly change the field of regenerative medicine. So I see human adipose stem cells as a light in the field of controversy. Despite all these controversies with stem cell topic, I still chose to explore human adipose stem cells because I have a passion for it, and even despite the potential negative results. Human embryonic stem cell research was challenged when it was first encountered, but scientists have no choice but to look under that microscope and describe it for what it is. However, scientists have yet to put a label on human adipose stem cell research, and I'd like to contribute to this to help it put it in a positive light rather than a negative light because of the forms of treatment that it could provide. So for all of you guys who do have ideas that you think are stupid or maybe just don't have much support for it, I say just go for it. Oftentimes, you can look under that microscope and describe it for what it is rather than what others see it as. Thank you.